Hi there, everyone. We'll go ahead and get started. I think some people will still trickle in. I'm Andrew Anfora. I am the Senior FAS Manager for Universal Sequencing. This is another one of our weekly Q&A sessions. Just to remind let those of you that are new or those of you that are returning, reminder, we are trying out a format now where every other week will be a general presentation and interspersed with more specific applications. So this week is going to be a brief general overview of our technology before we open it up to question and uh, answer. So in addition, we have today on the universal sequencing side, we have myself, Dr. Aaron Levy, our Chief Commercial Officer, Tom Chen, our CSO, and David Nordell, our Inside Sales Manager, are all uh, on as well. And just as a reminder, your mics are currently muted if you are not a presenter. So feel free at any time to type questions into the chat window, and Tom or I will answer them. Uh, and I think with that, I should probably get started so we can get to question and answers after the brief overview. So just a reminder of what universal sequencing's TELSeq is. It is a whole genome sequencing focused library prep kit that is a simple one tube library prep. It functionally converts a short read Illumina sequencer into being able to produce long read type data, including molecules upwards of 100 kb in length, with very low input, depending on the genome size being studied, anywhere from sub-nanograms to 5 nanograms of DNA input is required. And not going to go into really much detail in today's remarks, but you can use TELSeq for a number of applications, whether you are de novo assembling small, medium, or large genome. And you can also phase diploid organisms to aid in SV detection. And we have recently released some targeted approaches to phase more targeted regions down to PCR amplicon size fragments. So I'd like to give a overview of just some general background and a quick run through of our technology before opening it up to question and answer. So what's depicted on this slide is a high-level comparison of NGS platforms. And there's a cartoon representation of a diploid organism where you can see one chromosome labeled in blue and the other in red. So working from top to bottom, we see that short read platforms, which for the purposes of this talk are primarily Illumina sequencers, are capable of generating highly accurate, cost-effective reads of 1 to 500 base pairs in length. The a limitation of short read sequencing is that it is difficult to prepare a robust de novo assembly and is practically very challenging to phase haplotypes unless variants of interest are within 500 base pairs of one another. One solution to this limitation of short read sequencing is true long read sequencing, such as PacBio or Oxford Nanopore, where molecules of greater length are able to be assessed. So in the case of PacBio, the user has the option of choosing highly accurate reads of around 10 to 15 kb at a higher cost per base pair acquired, or slightly longer reads with lower accuracy. And these longer reads do enable more robust de novo assemblies and some degree of phasing of SNPs of interest. So to bridge the limitations of uh, cost, quality, and robustness, we have created a linked read reagent. I'll go into the mechanism a little more in the next slide, but at a high level, we take intact very long DNA fragments and apply a barcode pre-fragmentation so that we can look back at the short read library fragments and assign them to a molecule of origin. And we uh, in the slide, we indicate we can capture molecule information up to 150 kb in length. Really, the major limitation is the length of DNA going in. We can certainly capture molecule lengths greater than 200 kb if there are such molecules present in a data set. So not only are we approximating what you can do with long read sequencers, we are actually able to capture larger molecules than PacBio systems are able to do with high accuracy. I'm not going to go into too much detail about the mechanisms of our library prep for the sake of time. I do want to mention that our workflow is very straightforward and does not require any exotic equipment. All the manipulations take place in a PCR tube and utilize a magnetic stand. Key points of our protocol are that we use beads that have unique 18 base pair uh, uh, barcodes, and this gives us a theoretical library of approximately 2 billion barcodes to label molecules. We will mix these beads with a transpososome complex and long DNA. Long DNA for us, for most workflows, should be molecules greater than 10 kb in length, ideally greater than 40 kb in length. We will mix the transpososome, bead, and DNA, and it will form the complex you see here with the transpososome binding the barcode site on the bead, as well as the super long DNA molecules. We then add a second transpososome for tagmentation of the DNA. We break and wash the DNA fragments, and the DNA fragments that contain both transpososomes in the proper orientation will be amplified by a PCR reaction which adds the P7 and P5 tags to make it a Illumina compatible library. So our workflow is very quick and requires minimal hands-on time. From a purified high molecular weight DNA to sequenceable library takes 3 hours. So we are very competitive in terms of turnaround time of our workflow. Once somebody is familiar with our workflow, it would be possible to start with biomass DNA isolation and still be able to set up your sequencing run by the end of a single workday. I'm not going to go into much detail on our software, just at a high level. Uh, we provide free software tools for uh, QC and denoising of our data. And we currently have a de novo assembly tool for small and medium-sized genomes available, and we also have a phasing tool available. These software tools are freely downloadable as Docker and Singularity images from our website. Additionally, the QC tool and the de novo assembly tool are currently available on Illumina's base space. And we are in the process of beta testing the phasing pipeline, which will also be available on base space in the very near future. Additionally, our data type can be converted into a format that is compatible with 10x Genomics linked read tools. Our data types are similar, so many people are interested in continuing to use Long Ranger and Supernova, and we have a conversion tool to enable that workflow. Additionally, after running our QC pipeline, you can take our data and do something else entirely if you think you have a better idea than is depicted here on these slides. So really just before opening it up to questions, just reiterate that we are a very rapid workflow. So 3-hour library prep that requires very low input. So if we are looking at a microbial genome, it would be a sub-nanogram quantity of DNA. For a large genome, we would need 3 to 5 nanograms of DNA. Our assay is able to preserve super long DNA fragment lengths. Uh, the primary limitation is going to be what you're actually able to successfully isolate. We can routinely capture molecular information greater than 100 kb in length. And our software is free to enable use of our assay. We really don't have any hidden costs with our workflow. All you're going to be purchasing is the library prep reagent. And I think that concludes prepared remarks. So if you have questions you would like answered, Tom or I will address any technical questions, and Aaron and David can address if there's any sort of ordering logistics questions. So the first question I see is, what extraction kit do you recommend to obtain high molecular weight DNA? There are a number of commercial kits out there. One that I know off the top of my head is the Gentra Pure Gene from Qiagen that can support a wide number of tissue types. We have not specifically tested anything other than the homebrew method that Tom is calling out, but we're looking to uh, continue to expand guidance. I think the main thing to look at for a high molecular weight extraction kit is to see how the vendor is defining high molecular weight and see if you can get some type of gel image representation of what the kit is providing. High molecular weight DNA is a term that means different things to different use cases. So as long as a kit can produce the majority of its DNA fragments greater than 40 kb in length, we would expect it to work well for our application. So Filippo, I think we'll reach out to you for that one offline. It so briefly, the question is around adjusting the tagging enzyme to adjust insert size. So a lot of our protocol is modifiable, and we have varying degrees of performance expectation for those modifications. Um, Generally, reach out to either our tech support email, myself, or Tom for those types of questions. There isn't a one size fits all answer. In general, we get the question a lot about increasing the insert size for our library. Our default workflow will produce a 200 base pair insert. We find that is sufficient for most applications. We don't get these very long fragment information by having a large insert. We get it by linking the library fragment. So really, we do recommend trying to generate data using our default workflows first and before going ahead and increasing insert size. For the whole exome protocol, we do have a protocol that is targeted towards whole exome. Just to clarify, we have our standard library prep protocol, and we would expect that to not produce enough DNA for a hybridization capture. That should yield around 2 to 4 nanomolar library concentration. We do have a protocol optimization for whole exome, where it is effectively adding 3 PCR cycles. Again, I think that would be a good one using it. Okay. So for that question, so it sounds like if you are using the exome and still getting low yield, that would also be something. Email our tech support. That's going to be a little bit of a longer discussion to check on things and probably want to see some data. For marine creatures or insects, is there any preference for using the head or tail to create better samples? That is an excellent question. So for insects, whenever, if you're doing an insect isolation at least, and that's where I have the most experience between the two, if you have to use an adult insect, use the head. That's going to have more DNA and less exoskeleton. Really best case scenario for insects, if you're able to harvest uh, larvae or pupae, really the younger the organism, the easier it's going to be to isolate DNA. Pretty much any whole organism DNA isolation, if the organism doesn't have blood you can isolate, you generally want to use the youngest biomass available, tends to have the most actively growing cells. So really the highest highest mass of DNA per unit mass of organism.
Yeah, I think previous what I want to mention is that it really depends on application. For human sample, for sure we want to get a really good high molecular DNA, which is 40 kb. We consider is a short. And however, for some microbial application, for the especially for individual microbial sequencing, ideally the longer the better. However, if you get around 20 kb, it's good enough for our application of our sequencing.
Okay, so the next question is, are there any temperature restrictions when doing the DNA extraction? That would probably be highly method dependent. So it would really depend on the method that's being used. Obviously, you wouldn't want to heat it to the point that it would catastrophically denature. Beyond that, I don't know that there's a universal recommendation for extraction that it would be method dependent. Many are room temp, but not all are. Depend on if there's an enzymatic step or not. I don't know. Anything else to add on that, Tom?
No, yeah, it's usually the room temper up to 37, quite common.
So one more question. So is there a negative effect of doing the library prep if the temperature is not 37 as recommended, but 2 to 3 degrees below? Depending on the step, it is important that some of our steps are actually carried out at 35 degrees. So depending on the step in the workflow, most of our incubations with the rotation of the beads are actually 35 and not 37.
We're still investing further to really figure out what's the food impact. So at this moment, we still recommend people doing the 35. Same thing, if you go down 33, it's not a significant different in terms of 35 to 33. By the way, we do see that with our current condition setting, we'll be testing anywhere from from 32 to 39. At the end, we'll be settled down to 35. It is appeared more robust.
Great, thank you, Tom. So just that was same person asking some of the yield questions. So I think if you think you're seeing a yield issue and the temperature was 2 or 3 degrees outside of our recommended 35 set point, that would absolutely be one of the first things to go back and try and repeat within the recommended 35 degree incubation temp.
I think maybe we can put the last slides about some of the references we have on the website and recent videos, the YouTube. You mentioned a couple items.
All right, yeah, let me. So there are, just to close out, here are some additional readings. If you weren't familiar with some of these resources, and this list will continue to grow. We can probably start updating a few more items on here, but here's some great background reading if you're interested and want to know more, or had a question that you were afraid to ask. You might have an answer in some of these links. All right, well then, thank you very much for everybody who's in attendance, and I think this wraps this week's Q&A session.